Thank you. So the next point, I talked to you about the hardware. The next point is the algorithm. So it's clear that the future for neuromodulation in pain relief belongs to closed loop and ECAPs. Um, so all of the systems recently has to not only stimulate, but also record and analyze the signals from the body. But the problem is that doing this from the epidural space, we are receiving a kind of blurred image of the signal. Signal is noised, signal is full of artifacts, which scientists describe. Scientists and companies are working on the algorithms, sophisticated, which require a lot of energy to perform this computation. So we just realized that something has to change. Someone has to bring new approach to deliver system which will be available to do closed loop on the in the good results. So here is our first in vivo result from sedated sheep. It was done about 3 months ago. And you can see a red line, which is epidural signal recorded from epidural space with usage of high developed market available lead, epidural lead. And the signal is almost flat. And the second blue signal is from our intrathecal electrode, in which you can see very clearly P1, N1, and P2 peaks of ECAPs. I would like to quote to summarize this part. I would like to quote one of the companies who are in the ECAP and closed loop space. This is Saluda Medical, and they claim that epidural space is noisy, and we also see this in our tests. So now it's clear that no one system is ready to perform a closed loop approach on the market. And this is not only because of the recording, but also on the stimulation. The lead which is placed epidurally is far from the target, is surrounded by fat tissue, blood vessels. There are many tissues in between, like dura mater. So in our approach, we miniaturized our electrode. We moved electrode to the intrathecal space, which is filled of cerebrospinal fluid, which is also very important in our approach. And because of this, we received very good data, also very good results also in the stimulation. And these are these results which we are recently grabbing from 7 sheep. This is the chronic trial, 3 months. This is the second week of the trials, and you can see here the epidural lead and the signal from the epidural lead, and the ECAP is coming on the level of 0.6 milliamps. Stimulation, and with our solution, epidural solution, the ECAP is very, let's say, beautiful, for me beautiful, beautiful shape, all of the peaks, P1, N1, and P2, and ECAPs is coming on the 0.2 milliamps. And even just yesterday, I was reported that now we have, when the body stabilized, now we have also results on the level of 40 microamps, so much lower current is needed to stimulate with intrathecal electrode. We did also simulations, and here you can see the difference between, these are not our simulations, this is from Huang, 2014. You can see the focus of the current and the deepness. And in the intrathecal solution. But also we confirmed it in our simulations, that the signal and the stimulation is much better focused. We can achieve the correct fibers much faster and on the lower current activity. I mentioned several times about the power demand. What we would like to do with this, because our system relates on much lower current, we would like to democratize spinal cord stimulation, miniaturizing IPGs. So the same what's happened with pacemakers. And here, as David Dinsmore is here, I also wanted to quote his post on LinkedIn, and we are acknowledged his words, and this is kind of guide for us what direction we should choose to prepare the final product, which will be accepted by doctors and by patients. Last thing which I didn't mention yet is scar formation. So in the currently used leads in the epidural space, we see that the leads are surrounded by scar, by scar tissue. We still need to grab more data, but at this stage, after several tests in intrathecal space, we don't see any fibrosis, which we can see in epidural leads. And the same results are coming from the intrathecal drug delivery systems, which are in use since 30 years, over 30 years. And also these catheters, when they are removed, we can't see on these catheters any scars. So it's also kind of advantage of being intrathecally. Of course, we are aware that our system is still not clinically validated, and the maturity of our technology is still emerging. So we are working with very good team who support us. These are the co-founders. We have also two very distinctive key opinion leaders who are working with us on the story, on the technology, and how to deliver it, and how to get acceptance from the neurosurgeon, from pain doctors. Yeah. I will not talk about this as Stefan told us about the market. I just can mention that these data are very not updated. So Tim, this is something which you could do, your team, to update these annual losses on the pain. Our goal is to move this market, this fractional part to this one, the largest one. So democratize spinal cord stimulation to be used much more often than now. The next steps is neurorehabilitation, spine-computer interface, Parkinson's disease, but now we are focused on the pain. We just finalized our preclinical stage. Now we are raising next round is we opened next round last September. We are raising to get approval from FDA for IDE. And to be first in human, our plan is to be next year, not 26, 27, in the human. And good news is that we have already lead investor. So we are looking for following venture funds to join this round and to support us, support development. Thank you very much.
All right, Pavel, that was a great presentation. Thank you. I'll open it up to questions, but I want to take the liberty of passing the mic to Tom. Would you be willing to answer the question? And I don't want to take away from your talk tomorrow, but in those early days of spinal cord stimulation, talk about, was there a discussion of intrathecal implantation versus conventional dorsal column? Anything you might have to share? And sorry to put you on the spot.
That's a tough, I don't know what to say, actually. It was the choice at the time to put the sew the electrode into the dura. So we were the platinum interface was on the subdural side, and that was our thinking at the time. We never considered epidural at all,
I know.
but it clearly was made a difference when the pain community, the anesthesiologist, could put the electrode in the epidural space. The acceptance of the community blossomed, from my perspective. My own time at that was my last time in pain. I said, I don't want to do this anymore because this is not, you can't measure this stuff. So that was more the experience that I had at that time. I'm not sure that satisfies you, but it certainly put me on the spot.
I did, and I apologize for that. But I,
Thank
you know,
Thank you.
I had the question, and Pavel,
Yes,
I'm sure other people have. Well, why hasn't anyone done this before?
I
And
will-
maybe it's because it's always been done this way.
Yeah.
We have one
Unfortunately,
more question.
I was not able to talk to Dr. Shili because we started these projects. I joined at the very end of his life. But yeah, it's very interesting. The history is very interesting.
So I'm Dave Dinsmore from Medtronic. Thanks for your presentation. Maybe a more directed question would be, what are your thoughts on managing the incremental risk to the patient associated with an intrathecal lead placement versus a conventional epidural placement? Thanks.
Great question. So what we see based on our preliminary results data on, of course, sheep, not human, but also we have several tests on cadaver, human cadavers. We see that intrathecal space is not as risky as people used to think. Especially, we have data from catheters, intrathecal catheters, which are not generating any huge side effects. The second, always I'm questioning about leakage, cerebrospinal fluid leakage, and we see in our tests that dura mater is healing itself in several minutes after implantation in this space. So, of course, many tests ahead of us yet, but miniaturization of this electrode helped a lot, and we will even miniaturize more the dimension. Now it's 0.8 millimeter. Our goal is 0.6, and we think that it helps a lot.
Okay, I'm going to throw one more question at you, just really to follow up on something Tom mentioned, and that is that by implanting epidurally, maybe we're able to recruit a larger implanter population, anesthesiologists, and not just neurosurgeons. But I'm wondering if there's different approaches. A couple of companies I wanted to mention that I think are here. So there's an Italian company called Wise. Luca, are you here? So here's a technology that maybe is interesting. I'm wondering if there's a potential partnership here where you can, deliver a lead percutaneously and yet have it turn into a paddle lead once it's implanted. So I'm just kind of throwing that out. The other company I wanted to mention that's here, and is Cardis, are you here, Kartik? At any rate, so there we are. So this is a company that's really innovating perfecting the implantation process. And so they have done some work in intraoperative monitoring to help the surgeon or the implanter deliver the lead in the most precise location so that that you're getting the best results. Now, obviously closed loop stimulation is important. You've made that point, but I guess what I'm getting at, are you open to potential partnerships with companies like that who might be able to the bottom line is make the patient feel better, to make that pain
Yes.
go away at a higher percentage than we're currently able to achieve today.
Yeah, my life purpose is to help patients as fast as possible. And in my first venture, which I led 13 years, I had huge team, 30 people, and we used to not collaborate with anyone. But now I know, I understood that only collaboration can accelerate this.
Yeah.
So we are collaborating with SIPCA, we are collaborating with Neobionica, we are collaborating with many other companies, Ripple, to accelerate this work. And this is also why just only after 2 years, we are ready to biocompatibility and safety tests. It's super fast. And our plan is to be next year in humans. So this is only the reason why we can do this, because we are networking.
Okay, well, thank you very much, Pavel.